We're going on all year, June 2016, Biology, Unit 1, Paper 2. Here we go. Question 1a. Figure 1 shows the formation of sucrose. So here we have two molecules, glucose and fructose, and this is going to form sucrose due to a reaction R, and this is also going to let out a substance X. Name the reaction label R and the reaction product label X in figure 1. Reaction R is called a condensation reaction, and the substance X is water. So let me show you how this is done. So here we have a molecule of glucose and a molecule of fructose, and you'll see here, well, what is supposed to be here, by the way, although it's not depicted, well, these would be hydrogens. Sometimes you don't have to draw them, and these are carbon rings. So where you see it have each little junction here, each one here is a carbon, and this is how this bond is going to look. So you're going to see these hydroxyl groups, these OH and HO groups on the ends. Think of it like the ends of the two sugar molecules. Now what's going to happen here, this group and this H, imagine like this is going to combine here. And if you think about it, this is two H's and an O, so this is H2O. So this is actually losing a water molecule. But you're going to see that the oxygen over here, this is still left. So what's gonna happen here, and I'll just kind of take it out from here. So imagine like this oxygen is here. Well, there's gonna be a bond between these two, and the oxygen is gonna link them together. And well, because this is between two sugars, this is called a glycosidic bond. The prefix glyco relates to sugars, so for example like glycogen, and this is called a condensation reaction or a dehydration synthesis reaction. Our next question here, in the box below, illustrate the full molecular ring structure of the glucose molecule involved in the reaction in figure 1. Here you can see the molecule, but let me take you step by step on how to draw it. So this is the skeleton for a glucose molecule here. It is a hexagon. And the first, let me say that we start here. This is going to start actually with an oxygen, but it's going to be surrounded by carbon. So here is going to be a carbon all throughout. Now there are 6 carbons in glucose, because glucose is written as C6H12O6 with those numbers in subscript, of course. So there's 6 carbons, 12 hydrogens, and 6 oxygens. So that means that the 6 carbon is actually on the outside here. The 6 carbon is written as CH2OH. Now you can actually number them 1, 2, 3, 4, 5, 6 if you want. Now you're gonna see us, I say here, remember ho, 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 ho. And this is the reason why I want to remember that, because that will be how the placement of the hydrogens and the hydroxyls, the OH groups, are going to be. So on carbon 1 is going to be HO, H, and then OH, HO. Now remember, carbon must always be bonded, or must have 4 bonds around it. So for example, for this first carbon, 1, 2, 3, 4. The second one, 1, 2, 3, 4. Now remember, double bond would count as 2. But look at here, ho, ho, O. So on carbon 3, this is going to be O. And the fourth one is going to be ho. So I still have 1 more to put, because as I said, carbon must bond to 4 things. So this last one here, there's 1, 2, 3 to this carbon, and on a hydrogen here. So this is 4. And this is a glucose molecule. Ho, 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 ho. And the last one has CH2OH on it. Part 3. With reference to the molecular structure of sucrose, explain why sucrose has an advantage over glucose as a transport sugar in plants. But we would have seen previously that a sucrose molecule is a glucose and a fructose bonded together. So sucrose is a disaccharide, which means two sugars. So sucrose is less reactive than glucose. It is a non-reducing sugar and a disaccharide. So glucose is a reducing sugar. Think of that as simply meaning the bonds are a little weaker and they can easily be broken. Then glucose is much more reactive. Sucrose, on the other hand, the bonds take a little more energy to break, so it is less reactive. It wouldn't get used up as readily as glucose. So it's better for transport, basically. It wouldn't be readily absorbed into cells as fast as glucose. It is a more efficient energy transfer and storage molecule. B. Draw a simple label diagram to illustrate the fluid mosaic model of a plasma membrane structure. So you'd have to annotate. You just have the label is 5 marks. So what you want to have here is a phospholipid bilayer, and these will have phospholipids, of course, as a bilayer, so there's groups of 2 of them, and the phospholipids are going to have phosphate heads, and these phosphate heads, remember, hydrophilic or water loving, and they're going to have fatty acid tails, which are hydrophobic. So these are more going to be facing along the inside. Hydrophobic meaning that they would not really be in contact with the cell cytoplasm or the cell exterior, so they'll be hidden away. Kind of think of them like they're hiding in their own room. What you're also going to have to put here are some proteins. You can put channel proteins, carrier proteins, and these would be intrinsic proteins, which means that they span the entire width of the bilayer. You would also have cholesterol. You could just have a few molecules of cholesterol, which would be between the phospholipids. What those do is help maintain the fluidity of the bilayer. You would have some glycoproteins or glycolipids, which would be attached to either the bilayer itself or the intrinsic proteins. You can also add some peripheral proteins or extrinsic, which means that they don't span the entire length of the bilayer. The perfect way to think of a fluid mosaic model is think of there's a sea of phospholipids with little protein icebergs floating about. Once you have everything labeled here and drawn appropriately, you should be able to get your 5 marks. Part 2. With reference to the fluid mosaic model, explain how large polar molecules can pass through the membrane. Polar molecules means that they have a charge on certain areas of the molecule. So that charge there usually prevents the molecules from passing through the lipid bilayer. So for example, water is a polar molecule because it has the oxygen has a negative charge and the hydrogens have a positive charge, and because the inner part of the bilayer is hydrophobic, it sort of limits how much water could go through it. So polar molecules cannot easily cross the lipid layers. Polar molecules cannot interact with the hydrophobic regions. These would be the fatty acid tails. And usually movement has to occur through the channel proteins. Think of channel proteins as little tunnels that would allow passage of larger molecules like glucose and mRNA. Or carrier proteins. Carrier proteins, usually what would have to happen is that energy molecule or some other molecule or ion would have to bond it to open the gate. Think of these as gated channels. Channel proteins allow facilitated diffusion, and carrier proteins are gated channels that allow active transport. The use of ATP, usually conversion of ATP to ADP to allow passage of molecules against a concentration gradient. That means from a lower to a higher concentration. Question 2. A1. Using a simplified diagram of a small section of RNA, ribonucleic acid, describe the structure of RNA. You don't need to have the chemical composition of the components, which means that you don't need to actually draw all the carbons and oxygens and hydrogens on the sugars. You just need the basic outline of it. So first of all, how is RNA different from DNA? RNA is a single-stranded nucleic acid. Well, DNA is a double helix, it's double-stranded, where you would have one of the strands running anti-parallel to the other one. RNA is just a single-stranded, think of it like a lighter molecule. And from the diagram, we can see that there are phosphates, phosphate groups, there are bases, nitrogenous bases, and there would be 4 types of bases in RNA. There would be cytosine, uracil, guanine, and there wouldn't be any thymine, right? That would be replaced by uracil. You would have adenine. So usually adenine would bond to thymine in DNA, but in RNA it would bond to uracil. So RNA is single-stranded, is comprised of nucleotides and ribose sugars. So these are ribose sugars. These pentagon structures. Now in DNA, this would be a deoxyribose sugar. The only difference between deoxyribose and ribose is that deoxyribose lacks a certain oxygen molecule. So ribose sugar is linked to phosphates, and these bonds that you're seeing here connecting the ribose sugar to the phosphate, this is called a phosphodiester bond. So this is going to kind of be like a ladder of molecules here forming this nucleic acid. Part 2. Comment on the role of mRNA in eukaryotes. Of course, mRNA stands for messenger RNA. What mRNA does is that mRNA is built from a DNA molecule. The DNA unzips and an mRNA molecule is synthesized and elongated from reading the code on one of the DNA strands. So the mRNA is like a complementary strand to the DNA. The main difference is that RNA has uracil and DNA has thymine. The other main difference is that, of course, mRNA would be single-stranded. So mRNA is basically a copy of instructions that is being sent to ribosomes, so that the ribosomes could think of it like they can translate those instructions and build a protein by allowing amino acids to link to each other. So mRNA is sent to ribosomes in order to synthesize polypeptide chains, basically a string of amino acids, and proteins from genetic instructions. It does this by copying a complementary code from a DNA template and using ribonucleotides A, U, C, and G. There's no need to really get into the full description or transcription here, because and translation, I guess, because this is only 2 marks. Okay, so let's look at B. There's a lot to take in here. A plant geneticist is investigating the inheritance of genes for bitter taste, and this is given as a dominant allele, capital SU, an explosive rind, and this is given by a recessive allele, common E, in watermelon. Explosive rind is recessive and causes the watermelon to burst when cut. Non-bitter watermelons are as a result of homozygous recessive allele, common SU. So that means for the watermelon to not be bitter, it would have 2 of these SU alleles, common SU. The geneticist wishes to determine if the genes sort independently. A test cross is done between a bitter, non-explosive plant and a plant that is homozygous recessive for both traits. And they say table 1 shows the offspring. So let's look at how we're going to do this here. Well, first of all, let's look at the non-bitter explosive plant. Now they said that bitter taste is basically dominant, right? So the bitter taste would have the capital S in it. Explosive, they said, is recessive, right? Now non-bitter watermelons are as a result of homozygous recessive allele, common SU. So for something to be non-bitter, it would have 2 common SUs. And for something to be explosive, they say homozygous recessive, this would have to have 2 common E's. So in fact, anything that is explosive is going to have 2 common E's, and anything that is non-bitter is going to be 2 common SUs. Now, for something to be bitter, it has to have at least 1 dominant allele. So in this case here, this has to have at least 1 capital SU. Now, because one of the plants was homozygous recessive, as a result, one of them would be dominant and one of them would be recessive in terms of inheritance. So for the bitter one, you would have at least 1 dominant, and then you'd have 1 recessive, so it'd be heterozygous. And for this one, it'll be the same thing. For the non-explosive one, again, same thing. You would need at least 1 dominant allele, and because it was crossed with a recessive plant, you would have 1 dominant and 1 recessive. So non-explosive, you're gonna have a capital E and a common E. Same thing here, a capital E and a common E. Okay, let's look at this question here. Suggest a null hypothesis for the test cross in B. Alright, so just briefly go through this here. What a null hypothesis is to say that there's no significant difference between the ratio of observed results to expected, in this case here from a genetic ratio. But to give a simpler example here, I would imagine that you have a die and it was to keep tossing that die. And let me say that you toss that die. Now a die has 6 sides, right? And you was to toss that die 60 times. Now, what would you really expect to happen? What would be the probability that you'd expect? Well, if you toss it 60 times, you would expect that it would land on 1 10 times, 2 10 times, 3 10 times, 4 10 times, 5 10 times, and 6 10 times. Now, we know if we do that in reality, if we toss a die 60 times, it's not going to land exactly 10 times on each. In fact, those numbers may be slightly different. But let me say that I was to roll this die and it was to land on 50 times out of 60. And the rest of the times the rest of the sides it only land twice. Well, it means that whatever is happening is not due to random chance or random probability. It means that it could be a loaded die, right? It could be what somebody is used to cheat. So basically what a null hypothesis is used to do in statistics and with a chi-square, it is used to determine whether there are any external factors that are playing some kind of significant role in affecting the results of an experiment. And if there are, you would have to double check the experiment and see if you could remove those factors. So in this case here, we want to see if, in the case of these watermelons, if it's just due to the genes or if it's due to some other external factor causing the watermelons to be explosive or bitter. So the question here is for part 3. A chi-square test is conducted on the results of the test cross. Table 2 is incomplete for calculated values. Complete table 2 by writing the missing values in the relevant spaces. So this would be our table here, and I'll explain how we did this just now. And this is what the table looks like for chi-square distribution. Now, this table here, it might look very scary, but there's really only one part of it we have to look. Now, you'll notice that on the table we have what we call non-significant. So this is where there's not really a significant difference. And here we have a significant difference. Now, the threshold between these two here is this value here, what we call the critical value of 0.05. So it's really only this column here we want to look at. But which row are we going to look at? Well, remember there were 4 different phenotypes from the table. Right, there were 4 phenotypes. So degrees of freedom, to calculate that, we need to take the number of phenotypes and minus 1. So there were 4 different phenotypes from the table. So our degrees of freedom is going to be 3. So with 3 degrees of freedom, with a p-value or critical value of 0.05, we need to make sure that our value for the chi-square is less than 7.82, because if it's less than that, this would be being in the non-significant region where there would not be a significant change. So we want to make sure that it doesn't have that much deviation. 7.82 is the maximum chi-square value we could get. So let's work it out. So we're back to our table here. Here we have our 4 phenotypes, and these are observed numbers: 88, 68, 62, 81. Now if we was to cross the two plants from before, the expected ratios that we were going to get was 1 to 1 to 1 to 1. So the thing is, if we add up all of these here, 88, 68, 62, and 1, we could see we had a total of 299 plants. So if we do a ratio of 1 to 1 to 1 to 1, basically a quarter of that, and we was to round it off, the expected would be well, at least right below 75, but we could round off to 75. So it would be 75, 75, 75, 75. Now to work out the chi-square value, we would have to use this formula here. The observed minus the expected squared divided by the expected. So in this case here, if we say 88 minus 75 squared, and then divide that by 75, we will get our value here. So let me show you what I mean. If we take 88 minus 75, that will give us 13, and of course 13 squared is 169, and we was to divide that by the expected value, which is 75, we will get 2.253 recurring. We could just round that off to 2.25 as you see here. Just to test it for one of the other values, let's look at the second one, where our expected value is 75, our observed is 68. So we're going to say 68 minus 75, and that's going to give us minus 7. So of course, you know, minus 7 squared is still 49. If it's square negative number, you get a positive number. And if we divide that by the expected value, which is 75 in this case, you're going to get 0.653 recurrent. So you see this, we basically doing the correct thing. So in order to get the chi-square value, what we have to do is find the sum for each phenotype. So 2.25 plus 0.65 plus 2.25 plus 0.65. When we find the total of that, we are going to get 5.8. Now if you remember what our maximum value would have been from the table, we didn't want our value to surpass 7.82. So that means that we are actually in the safe zone here, because 5.8 would be somewhere between these two numbers here, and this is in the non-significant region, which means that there's no significant change, or no significant difference, I should say. So to answer our question here for 2 marks, using the table of probabilities provided below, and with reference to the calculated chi-square value from table 2, evaluate the validity of the hypothesis. Well, the chi-square value 5.8 is less than the critical value for 3 degrees of freedom, and the critical value we would have observed is 7.82. Thus, the null hypothesis will be accepted or fail to be rejected, since there is no significant difference between the observed and expected values. Basically, what this means is that it is within the realm of possibility, or probability I should say, that any changes there that between those two values are purely due to random chance, due to genetics. It is not due to some kind of outside influence that is causing the watermelon to be bitter or explosive. That's purely due to genetic chance. Question 3a. Figure 2 is a photomicrograph showing a cross-section of a mature anther. And here we could see the photograph of the mature anther. Now this is what your drawing would look like. What we have to do is to make a plan drawing of anther in the figure 2 and use annotated labels to identify 2 tissues. This is what your diagram would look like, your plan drawing, sorry, and you'll see that there are 6 labels here, but any 2 of these would do. So 1 is the epidermis, which is the outer protective tissue of the anther. 2 is the vascular bundle, which is made up of the xylem and phloem, of course, for water, mineral, iron, and sucrose transport. 3 is the fibrous layer, and this allows for dehiscence of the anther. Dehiscence means like the drying out and the splitting of the anther so that pollen grains could get released. 4, so this inner layer here, this is called the tapetum, and the tapetum is it provides nourishment for the pollen grains. within the anther itself, within the chambers. 5 is a pollen grain, which contains male haploid nuclei, which are ready to deposit onto the stigma of a flower to grow the pollen tube and eventually to fertilize the egg. And 6, this is the connective tissue, and this basically is kind of like the collagen of the anther. It keeps the support and the shape of the anther. Any two of these would work. Make sure that for your plan drawing that you don't put cellular details, that the outlines of the layers are clear and neat, probably neater than the diagram that you're seeing here. You also want to make sure that the lines are clean and continuous, and that it is representative of the photomicrograph. And finally, you want to ensure that you have a title. The title I have here is Plan Diagram of a Cross-Section of a Mature Anther. The magnification could be in relation to the size of the photomicrograph. So this would be your drawing size divided by the micrograph size. Remember, there's no units for magnification. Proper drawing, 2 annotations, title and magnification is what's going to give you 6 marks. 3b, figure 3 is a diagram of a section through the placenta with the umbilical vein. What you have to do here is to identify the structures labeled A, B, and C and indicate whether each is of fetal or maternal origin. Write your answers in the following table. So just to label it here. So A is going to be, so you're going to see that these are like finger-like projections that is coming from the umbilical cord, from the umbilical vein. So these are called chorionic villi. We could put it singular, chorionic villus, and this is of a fetal origin. What these do is that via facilitated diffusion and simple diffusion, this is going to either excrete materials to the maternal bloodstream, or is going to have an intake of materials from the maternal bloodstream. So for example, glucose and oxygen is going to go into here. Okay, so B, this is going to be what you call an intervillous space or intervillous sinus, and this is going to be of maternal origin. So everything that is surrounding the chorionic villi, everything here is going to be of maternal origin. So this is where the maternal blood is going to come in and is going to have contact with the fetal bloodstream. Now remember, the maternal blood doesn't directly flow into the fetal bloodstream. All it does is exchange materials through these membranes here. Finally, for C, this is going to be the endometrium or the uterine lining, and this is also going to be of maternal origin. This is where the implantation of the embryo is going to occur, and via the umbilical cord and the placenta, there's going to be exchange of materials and storage of materials and nutrients that's going to allow development of the embryo into a fetus finally, until it could become a baby. So here we have the 3 structures once again. A is the chorionic villus. This has a fetal origin. B is an intervillous space or sinus. This is maternal. And C is the endometrium or the uterine lining, and this is going to be maternal. Part 2, outline 2 functions of the placenta. Well, we have 3 here. Any 2 of these would work. The placenta is to allow exchange of substances, for example, gases, nutrients, and removal of waste products. 2, the delivery of maternal antibodies to the fetus. This would enact natural passive immunity, so that this is kind of like to kickstart the fetus's immune system. And 3, it acts as a barrier for larger materials and allows for regulation of maternal and fetal blood pressure. Larger materials would be like hormones. One want hormones from the mother going into the fetus's bloodstream. Part 3, comment on the importance of the amnion during fetal development. In your comment, give 2 effects of the reduced levels of the amniotic fluid. So we have 2 here is 4 marks. So for each one, we want to say, well, what will happen if there's a reduced level of amniotic fluid? So the first one is that the amnion supports the developing fetus against gravity, basically holds it in place. It also acts as a shock absorber. Shock absorber means that, we say, for example, if the mother was to get hit in her belly, it would absorb the force from that hit, so it basically limits injury to the fetus. So if you have low levels of amniotic fluid, this would lead to an increased risk of physical injury occurring in the fetus. It would also lead to the fetus not being held in place, so support and development might be impeded. 2, the amniotic fluid acts as a temperature buffer because most of it is water. And water has a high specific heat capacity. What does that mean? That means that water is able to absorb a lot of heat energy before it actually changes in temperature. So it helps to regulate temperature. Reduced levels of it can lead to high temperature fluctuations in the fetus, which you don't want, because there's a lot of enzymes that are now starting to be formed and developed in the baby's systems. So you don't want those being affected. Question 4a. According to the endosymbiotic theory, organelles such as mitochondria and chloroplasts are thought to have evolved from prokaryotes. Explain what is meant by the term endosymbiosis and how it relates to the origin of these organelles in eukaryotes. This is 3 marks. Now to break down the term endosymbiosis, endo means in, and symbiosis, kind of like, with symbiotic relationships, means that there's a relationship between 2 organisms that usually could be described as mutual, which means that they benefit from each other, or at least one of them benefits from the other. So endosymbiosis occurs when one organism lives within another. It should be lives within another, and both benefit from each other, right? So it means endo means in. Endosymbiotic theory. So just to give you a brief overview of this, what this is basically telling you is that very, very early on, at the beginning of primitive life on Earth, they said that all of it would have been prokaryotic, and said that there were very early prokaryotes that would have gotten the ability to respire, and those would have been very early mitochondria, what would become mitochondria now. And there were also prokaryotes that would have had the ability to take in sunlight and convert that solar energy into chemical energy. And those prokaryotes would have eventually become chloroplasts. So what the theory describes is the origin of organelles such as mitochondria and chloroplasts as very early prokaryotic organisms. And what happened is that those 2 very early prokaryotic organisms were engulfed by a larger host cell. And I like to think of it as if it was like it was giving them superpowers, or giving the host cell powers. Now the host cell had the ability to respire and convert, ATP to ADP, and have larger energy reserves, but the host cell also had the ability to convert solar energy to chemical energy by absorbing these 2 other prokaryotes. And that would have been one of the first origin cells for what would become a eukaryotic cell. So these enable the host to undergo respiration and photosynthesis eventually. Part 2. Discuss 3 lines of evidence in support of this theory. This is 6 marks, so whatever line of evidence you wanna compare the organelles mitochondria and chloroplasts to prokaryotes. They don't have to be ultra detailed, but the comparison should be there. So first of all, mitochondria and chloroplasts are similar in size to bacterial prokaryotes that exist today. The next thing here is mitochondria and chloroplasts have their own DNA, which is circular. We know that prokaryotes don't have a nucleus, or what you call a true nucleus. They have a nucleoid region, which usually contains circular DNA or plasmids. 3. Mitochondria and chloroplasts have their own ribosomes, and those ribosomes are 70S in size. S is a unit called sievert units, and that basically relates to, think of it as density, or how fast objects would sink in a medium, whereas eukaryotic organisms have 80S, or mitochondria and chloroplasts have their own. 4. Mitochondria and chloroplasts divide by binary fission, just as bacteria do, even though the eukaryotic cell may divide by mitosis. And 5. In the double membrane found in mitochondria and chloroplasts, the outer membrane is similar to eukaryotic membranes, but the inner one is more similar to prokaryotic. So that there is quite interesting. So what you want to do is you want to have 3 of these. I would suggest the first 3 are typically the easiest to remember. And you should get all your marks if you state that and compare it to prokaryotes. Let's look at B. So this is 6 marks. Enzymes are globular proteins and are highly specific in the reactions catalyzed. Explain the basis of enzyme specificity and include in your explanation a brief description of the globular nature of these molecules. First of all, you should describe, well, what is an enzyme? So you can get to see how exactly they are specific to their reactions. So enzymes are tertiary structures with 3-dimensional shapes. They consist of multiple polypeptide chains joined to each other via covalent, ionic, and hydrogen bonds. These allow the polypeptide chains to fold to form a very specific shape. Now, within that shape, you are going to have a depression or cleft called an active site. And that there is a specific sequence of amino acids. And that's going to form like a little indentation in it. Kind of like a keyhole that a key is going to fit into. So this forms bonds with complementary substrates like a lock and key. As an easy example would be amylase, which is an enzyme, and that bonds specifically with starch. And amylase would then hydrolyze starch, which means like break the bonds, break it down to form maltase. So sometimes the substrate fits into the active site, but the enzyme must slightly change shape to accommodate the substrate. So this is called an induced fit. So sometimes they will fit like a lock and key, where the enzyme and the substrate will form an enzyme substrate complex, which wouldn't be that different from before. But sometimes it's kind of like a hand in a glove, where the glove would sort of fall over the hand and change shape just a little bit to accommodate the hand when the hand fits into it. And the enzyme temporarily warps, right, or folds a little bit just to accommodate the substrate. Let's look at question 5a. Evaluate the use of genetically modified organisms or GMOs in agriculture. Include an explanation of the term genetically modified organisms. A genetically modified organism is one which contains genes transferred from another species or member of another species, or has had its genes altered or its genome altered. So what does this mean is that I like to use the common example of a glowfish, which is basically a zebrafish that has been implanted with a gene from a bioluminescent jellyfish or coral, and now it has the ability to glow when it contracts its muscles. Basically, you take a gene from one organism, transfer it to another using enzymes to do that. It's called restriction enzymes and DNA ligase. And with that, you can actually snip off a piece of DNA and glue it to the next piece of DNA to insert into another organism. That is what is called a GMO. Now GMO, they want us to focus primarily on agriculture. GMO crops typically have increased crop yields and provide greater food security, since crops would be less susceptible to insects. And this is also because some GMO crops can actually produce their own pesticides. For example, caffeine, which is not harmful to us really unless in very large amounts, but for insects, caffeine is fatal. So some plants can be infused with a coffee plant gene to produce caffeine, and as a result, when the insects eat the fruits or the leaves or the stems from those, they die. So this reduces the need for more harmful pesticides, leading to less exposure to toxic chemicals to the crops and to the people eating those crops. Some GMO crops have enhanced nutrients to combat nutrient deficiencies in humans living in impoverished regions. Example would be golden rice, and golden rice is basically rice, like regular rice grains, infused with a daffodil gene, and as a result now it looks a little more yellow in color. These contain beta carotene to mitigate night blindness, kind of like vitamin A. Lastly, GMO crops may be able to grow in areas where they usually won't, because they could make them not drought resistant really, but they could better withstand drought, which means that they could have more succulent tissues to retain more water, or they could be frost resistant as a result. So this means that more crops can be able to grow in more areas, and they could grow in at different times of the year that they usually won't. This of course would lead to better profits for farmers. Part B. Explain the steps involved in the use of recombinant DNA technology for the production of human insulin from bacteria under the following headings. So you have to focus on gene isolation and gene insertion into a vector. So just to give you a little backstory here, very long time ago, well, not very long, but a few decades ago, primarily insulin used to be obtained from pigs. And because pigs require so much resources to rear, and it was expensive to actually extract the insulin from it, insulin was in short supply. That would of course drive up the cost if you know anything about supply and demand. If the demand is high and the supply is low, it becomes very expensive. Now they realized that they could have used recombinant DNA technology. Recombinant means that again is genetic engineering. You're transferring one gene from one organism to another, so you combining the DNA. That's what it's called, recombinant. They realized they could have done that, and they could use something that is easier to mine than pigs, which is E. coli bacteria, which you don't need a mate for them to reproduce. They can reproduce in large numbers through binary fission, and you can keep them contained within a fermenter and just give them glucose and oxygen, and they'll be able to multiply and produce insulin. Now, how you do this is that you would have to take the insulin producing cells from the human pancreas, and you would need to somehow get that DNA, isolate that gene, wherever the gene that tells the body to produce insulin, and put that into E. coli bacteria. So how that is done is that mRNA, messenger RNA, is extracted from the cells. If you remember what messenger RNA is, that is the blueprint that is being sent to the ribosomes to make a protein. So this is extracted from the cells and incubated with reverse transcriptase. Reverse transcriptase is an enzyme that is going to convert the RNA to build a complementary strand of DNA. So remember, transcription was turning DNA, right, using DNA to build RNA. Reverse transcription is using RNA to build DNA. It's just the reverse. So the DNA strand is then incubated with DNA polymerase, and what this is going to do is going to allow more and more DNA polymers to be built, more and more DNA strands. So you started off with just one, but now you could have them in thousands. And free nucleotides to form the DNA helix. This is basically what is happening, is that you are building multiple strands of DNA, right? So you're isolating the gene, the mRNA, and you are building new DNA from that using reverse transcriptase. Secondly, we need to insert the gene into a vector. How this is done is that you use restriction enzymes. Restriction enzymes are like DNA scissors, and they are used to cut genes, and they're gonna leave little sticky ends. These sticky ends, think of them as they can bond with complementary ends, right, due to hydrogen bonding. So small circular DNA plasmids from bacteria are also cut with the same restriction enzymes. So it's kind of like you're cutting out a shape, and you're cutting out a similar shape in the bacteria so you can glue them together. So how are you going to glue them together? The plasmids and cut DNA are joined using DNA ligase to form recombinant plasmid DNA. So restriction enzymes are like DNA scissors. DNA ligase is DNA glue. To ligate something, by the way, is to like tie it together. Question 6a. Spore production is one type of asexual reproduction seen in fungi. Use an example in which you are familiar. Give an account of the process involved in this type of asexual reproduction. I should note in 2022, at the time I'm doing this video, they've actually taken out spore production and so on from the syllabus. So I'm not going to spend too long on it, but just for the sake of doing the question, I'm using the example of penicillin. So penicillin, the spores are called conidia, and those occur externally on the cells of production. Now, you can think of the hyphae or hypha. Think of that as like if the fungi had roots, the hyphae would be the roots. They would basically be the ones that would dig into substrate. So for example, you have bread mold and the little thin root-like white hairs that you see on it, that's the hyphae. That will be used to obtain nutrients for whatever substrate it is attached to. So the hyphae of the fungus is a chain of haploid cells, where new cells are produced at the end of the hyphae. Branches form at the tips of the hyphae and produce strings of spores via mitosis. The spores are protected by a waterproof covering and are spread in the air. So this is just giving you a beat by beat of how the spores are produced in this penicillium fungus, which is we used to make penicillin, so I should actually call this penicillium. Part 2. So this is still on the syllabus. You should still know the advantages and disadvantages of sexual and asexual reproduction. Fungi can also produce offspring by sexual reproduction. Discuss the conditions under which asexual reproduction is more advantageous than sexual reproduction. So remember, the difference between asexual and sexual is that asexual requires one parent, sexual requires two. Sexual requires the production of gametes, asexual doesn't. Also, with asexual reproduction, there's only one set of DNA being passed to the offspring, so each one is genetic clones of each other. Sexual reproduction, you would have genetic variation because is due to half the DNA being passed from each parent. Here we have is 3 marks, so we're going to have at least 3 of these points. So 1, when sexual reproduction takes much longer to produce offspring or more energy is needed to produce gametes. This is where asexual reproduction would work better, because let me say that you only have a few fungi around, most of them are dead, and you can't really wait for sexual reproduction to occur. If there's only one parent needed, it's going to occur much faster than with sexual, because there's no need to produce gametes, there's no need to fuse DNA and so on. Basically, they will bud, produce spores, and just spread like that. And this correlates to 2, if a large number of offspring are needed in a short time. The next thing here is if individuals are already well adapted to a stable environment, basically where you don't need natural selection being involved. If the individuals don't really need to adapt to the environment, if they're already well adapted, you don't really need genetic variation. They could all have the same type of DNA and all of them would survive just as fine. Lastly, if food material is abundant and can support a rapidly growing population. Basically, if there's enough food, more could come, more could grow. Any 3 of those, give it 3 marks. Our final question here is B. 6B, gonadal hormones coordinate activities of the ovary and uterus in human females. Give a brief description of the 3 main phases in uterine cycle, and for each phase comment on the role of gonadal hormones. Include the names of gonadal hormones. Alright, so the gonadal hormones are of course the ones that are made by the gonads, and they're primarily talking about estrogen or estradiol and progesterone. Not really talking about FSH and LH, because those are the pituitary hormones, and which are not produced by the ovaries in this case. We have 3 phases we're going to look at. We have the follicular phase, the luteal phase, and the menstrual phase. The follicular phase is basically when you have the development of the Graafian follicle, which contains the secondary oocyte. Now, during this phase, you're going to have the thickening of endometrium tissue. Basically, if the egg gets fertilized, it'll be able to implant onto that tissue, kind of like you're planting a seed in soil. You'll also have vascularization, which is the development of the blood vessels. So you're going to get more and more capillaries taking blood and oxygen to those tissues to be able to grow. Estrogen singles the endometrium to thicken. I guess a fun little fact here, or not so fun little fact. Because of the development of these blood vessels, when a girl undergoes menstruation later on, this is actually because those blood vessels get so constricted that oxygen cannot be delivered to the endometrium tissue, as a result causing them to necrotize or die. and that is when, you know, it would shed the lining, and as what would come out during menses, the, you know, menstruation. That's actually what causes the pain, that and the cramps. The second phase here is the luteal phase. So the luteal phase, basically this is when the follicle of the egg gets broken down into something called a corpus luteum. Corpus luteum just means yellow body. And what the corpus luteum does is that it secretes progesterone. Progesterone helps to maintain this thickened endometrium for implantation. So estrogen signals it for it to thicken. Progesterone sort of keeps it there, kind of holds it strong. If progesterone levels drop, that endometrium is gonna get the blood vessel is gonna get constricted, and eventually gonna get menstruation. Uterine lining is gonna break down. So if a woman is pregnant, the woman has to keep producing progesterone for prolonged periods of time so that the uterine lining doesn't break down. Except in that case, it wouldn't be via the corpus luteum. That will be via more the blastocyst, and the placenta will be producing that. The last phase that we have here is the menstrual phase, or menses. This is what is described. A decrease in progesterone levels leads to the breakdown of the endometrium. And this will occur if no fertilization of the secondary oocyte has occurred, because if that has not been fertilized, you will not get a blastocyst, you're not going to get an embryo and a placenta, so of course you're not going to get the continuation of progesterone occurring. So what you want to do is describe these three phases here, name the associated hormones, and you should be able to get your 7 marks. And that's it for this paper. Take care. Laters.